So it will be Lac Operon 2. In this lecture, we'll primarily discuss the catabolite repression. All right, this is the usual disclaimer, so I will not go into the details of this once again, that it is meant for you students and it should not be used for any other purpose, copied or posted on any site without my permission. And some of the pictures in this presentation, they have been taken from public domain websites to make the topic easier for you to understand. The credit for such pictures remains with the original publisher, and I have tried to acknowledge it everywhere. All right, so what we discussed yesterday. We discussed yesterday that Lac Operon is an inducible operon, right? And lactose is the substrate for this operon. It serves as an inducer. That is, it is only in the presence of lactose that operon will be switched on. We'll be using these words very often, switched on, switched off. So lactose is the substrate. Unless lactose is not there, for bacteria, there is no purpose synthesizing those enzymes which are required for the uptake of lactose and its catabolism. It will be just wastage of energy and resources. So unless lactose comes into the medium, those enzymes will not be synthesized. So lactose serves as an inducer. Those enzymes are called as inducible enzymes, right? Now, what is its status under different sets of conditions? So this was an interesting, what you can say, thing that we discussed yesterday. So let's see one by one. When lactose is absent, so by now you know it very clearly that the operon will remain switched off because there is no point synthesizing those enzymes which are required for uptake of lactose and its catabolism. Right? What happens when lactose present? When lactose comes into the medium, immediately, please remember those three things. That it should be flexible. That means it should be able to switch on from one place, shift from one metabolite to the other metabolite. So as soon as the lactose comes, it should be able to immediately switch over to metabolizing lactose, right? So the operon will be switched on and it does so very rapidly. Swiftness is also required. So the operon is expressed, or we call it as switched on, or you can also term it as turned on. Okay, so operon will be expressed and the bacteria will start utilizing lactose for its energy purpose. Okay, third thing that we discussed yesterday is, let's say there is a mutation in any one of the structural genes. We said that there are three structural genes in Lac Operon. Lac Z, Lac Y, and Lac A. So if there is a mutation in any one of those three structural genes, what is going to happen? That particular gene and its product, that means the protein it codes for, that is going to get affected. For example, if there is a mutation in Lac Y, okay, so only Lac Y gene and its product, that is permease, the protein that it codes for, permease enzyme, these two are going to get affected. Rest of the operon will work. If there is a mutation in regulatory gene, like I, if there is a mutation in regulatory gene, that is like I, what is going to happen? Expression of all the structural gene that it controls. Whatever structural genes like I controls, and it controls the three structural genes, the expression of all the structural gene is going to be affected. And those genes, irrespective whether lactose is present or absent, those genes will be continuously expressed. That means the operon will remain on. The operon will remain switched on. Okay, the technical term for that is the operon will be constitutively expressed, or the genes will be constitutively expressed irrespective of presence or absence of lactose. Because the Lac I is mutant, as a result of that, that repressor protein is going to be mutant and it is not going to bind to the operator region under whatever condition. Condition means whether lactose is present or lactose is absent. Now, we made things a little more interesting. So let's say if you make the cell partially diploid with a wild type Lac I. So original Lac I is minus, mutant, shown over here. Okay. What we have done, we have added a piece of DNA which contains a wild type Lac I. We introduce this wild type Lac I along with its promoter into the bacterial cell, which has original Lac I is mutant. So we made it partially diploid. What is going to happen? The wild type inducibility is going to be restored. That now the operon will respond to the presence or absence of lactose. That it will get switched on in presence of lactose, and it will remain off when the lactose is not there. What we call it as wild type. Original condition, original situation is going to be restored. In technical terms, it is called as wild type inducibility is restored. And why so? Because Lac I is supposed to be transacting. Okay, I introduced the word transacting yesterday. Lac I is supposed to be transacting, right? Let's say there is a mutation in operation